Hello everyone, I am Subhalakshmi. Welcome all of you to this channel. In this video, I am going to explain about 20 multiple choice question on the topic of reproduction and life cycle of fungi. Before we start the discussion, let's begin with a beautiful message. Success is not a one-time event, it is the result of consistent effort. So, stay true to your path and let go of what holds you back. Work steadily with humility, peace, faith, wisdom, courage, and determination, then success will follow you. Okay, see the first question. Which of the following is a holocarpic fungus? Option A, Ostilago. Option B, Agaricus. Option C, Syncytium. Option D, Sperotheca. Here the correct answer is option C, that is Syncytium. Depending upon the how the fungal thallus is involved in the formation of reproductive structure, they are divided into two types, that is holocarpic and eucarpic. Then what is holocarpic? Remember, in this category of fungi, the entire thallus gets converted into one or more reproductive bodies. Hence, the vegetative and reproductive phase can never occur at the same time. So, the holocarpic thallus does not show any differentiation on vegetative and reproductive structure. For example, yeast and Syncytium. Then what is eucarpic? Remember, most of the fungi are eucarpic. Here, only a part of the thallus is involved in the development of reproductive organs and remaining thallus remains vegetative. Okay, and in eucarpic fungi, vegetative and reproductive phases exist at the same time. So, the eucarpic thallus is differentiated into vegetative part which absorb nutrients and a reproductive part which forms reproductive structure. For example, pythium. Okay. And here we know Syncytium is a large genus of plant pathogens within the phylum Chytridiomycota that causes potato rot disease. Okay, and Syncytium is a holocarpic fungus. Then see the other option, that is Ostilago. Ostilago is considered as a eucarpic fungus. This means that the thallus is differentiated into vegetative structure and reproductive structure. So, the reproductive structure like the spores are formed by a specific part of the thallus, not the entire thallus. So, Ostilago is a eucarpic. Then in the option B, that is Agaricus. Agaricus is a genus of mushroom forming fungi containing both edible and poisonous species, and Agaricus is considered as a eucarpic fungus. This means that only a part of the thallus is involved in the development of reproductive structure while the rest of the thallus remains vegetative. Then in the option D, that is Sperotheca. Sperotheca is a genus of pathogenic fungi. Is known for its distinct reproductive structure indicating it is an eucarpic fungus. Okay. So here Ostilago, Agaricus and Sperotheca are eucarpic fungi, but here Syncytium is a holocarpic fungus. Then next question, Emericella is a perfect stage of option A Alternaria, option B Aspergillus, option C Amanita, option D Ascobulus. Here the correct answer is option B, that is Aspergillus. First you should know what is perfect stage. Remember, in fungi perfect stage means the sexual stage. Okay, so if a fungus producing asexual state is termed as anamorphic or imperfect stage, and the sexual state when present is termed as telomorphic or the perfect stage. Okay. Here the genus Aspergillus is well known for its pleomorphic life cycle, meaning it can exist in both sexual and asexual forms. So, the perfect stage of Aspergillus involves sexual reproduction where haploid nuclei fuse to form a diploid zygote which then undergoes meiosis to produce the haploid ascospores. And the sexual stage of Aspergillus nidulans is identified as Emericella nidulans. Okay, and this stage is less common and primarily found in specific environmental condition. So here Emericella is a perfect stage of Aspergillus. Okay, and here also the imperfect stage of Aspergillus involves asexual reproduction primarily through the production of conidia, right? And Aspergillus nidulans is the asexual or imperfect stage of Emericella nidulans. Okay, then see the other option, that is Alternaria. Alternaria is a genus of Deuteromycetes fungi. All species are known as major plant pathogens. For example, most common species, that is Alternaria solani, causes yearly blight of potato. Then in the option C, that is Amanita. Most of the species of this genus are toxic because it contains psychoactive alkaloids belonging to the Basidiomycetes group. Then in the last option, that is Ascobulus. Ascobulus is a coprophilous fungus, that is it grows on animal dung and the genus has a widespread distribution. Okay. Then next question, whose spores are found in option A Sperotheca, option B Poxinia, option C Mucor, option D Phytophthora. Here the correct answer is option D, that is Phytophthora. In the life cycle of Phytophthora, oospore are formed through the sexual reproductive process resulting from the fusion of an antheridium and an oogonium. And when conditions are right, oospore can germinate and release zoospore, which are the primary source of infection for plant host. Okay, so oospores are thick-walled sexual spores that crucial for the survival and persistence of the Oomycetes fungi. So here the correct answer is option D. And we know Phytophthora is a genus of plant damaging Oomycetes whose members of species cause economic losses on crops worldwide as well as environmental damage in natural ecosystem. Okay. Then see the other option, that is Sperotheca. Sperotheca is a genus of Ascomycetes fungi and oospore are not found in this group. Okay. Then in the option B, that is Poxinia. Poxinia is a genus of fungi that causes rust disease in plants and oospores are not typically found in the rust fungus. Okay. Then in the option C, that is Mucor. Mucor is very fast growing genus of fungi. Commonly it is known as bean mold, and remember the primary sexual reproductive structure is the zygospore, not oospore. Okay. So oospore are found in Phytophthora. Then next question, in which fungi branched hostorium is commonly found? Option A Poxinia menthe, option B Erysiphe polygoni, option C Mucor mucedo, option D Fusarium solani. Here the correct answer is option A, that is Poxinia menthe. What is hostorium? We know hostorium is a specialized small finger-like or spherical structure formed from the filaments of fungal body, and the primary function is to absorb the nutrients from the host plant. And remember, hostoria are variously shaped, being knob-shaped in Albugo candida, large and irregularly swollen in Pornospora parasitica, and branched in Poxinia menthe, which causes rust on mint plants. So, the branched hostorium is commonly found in Poxinia menthe. Then see the other option, that is Erysiphe polygoni. It is powdery mildew fungi, constitute a common group of obligate plant pathogens that causes powdery mildew in black gram and causing significant yield losses. Okay. Then in the option C, that is Mucor mucedo. Mucormucedo is a fungal plant pathogen commonly found in soil, dung, water, plants and moist foods. And remember, particularly this species of mucor is called common pin mold due to its pin-like sporangia. Then in the option D, that is Fusarium solani, it is an important soil borne plant pathogen which can cause damage to many agricultural crops. Okay. Then next question, the fruiting bodies of which fungi exhibit bioluminescence? Option A, Pleurotus, option B, Ganoderma, option C, Allomyces, option D, Mycena. Here the correct answer is option D, that is Mycena. What is bioluminescence? We know it is a natural phenomenon where light is produced by the living organisms. So here many species of the Mycena exhibit bioluminescence in their fruiting bodies, particularly Mycena chloropus, which is known for its bright green glow fruiting bodies. Okay. And some studies have also found that mycelia of this genus can also exhibit bioluminescence. It is the image of Mycena in daylight and it looks bright green color during dark. And remember, not all Mycena species are bioluminescent. A significant number including Mycena chloropus, Mycena corocata and others have been observed to produce the light. Okay. Then see the other option, that is Pleurotus. Bioluminescent is not a characteristic of this genus, and this genus is known for its edible fruiting bodies and other beneficial properties, not for the emitting light. Okay. Then in the option B, that is Ganoderma, it is a genus of mushroom known for their medicinal properties, not for bioluminescent. Then in the option C, that is Allomyces. Allomyces is a genus of fungi known for its unique life cycle and reproductive strategy, but it is not known for bioluminescence. Okay. So the fruiting bodies of Mycena fungi exhibit bioluminescence. Then next question, torula condition is primarily observed in option A Pejija, option B Mucor, option C Ostilago, option D Albugo. Here the correct answer is option B, that is Mucor. In Mucor, when it is allowed to grow on sugar rich media, it produces small spherical cells which are known as oidia. The oidia are thin walled like structure. Then after formation of oidia, it gets detached from the body of the mucor and increases by budding like in yeast. And this stage is known as the torula stage. And remember, the word torula comes from the Latin word torus, which means swelling, and this stage is commonly observed in some particular species of fungi like mucor and rhizopus when they are grown in a sugar rich medium. Okay. Then see the other option, that is Pejija. This genus is known for its cup-shaped fruiting bodies called apothecia, and generally they are found on the ground or on rotting wood. Okay. Then in the option C, that is Ostilago. Ostilago is a genus of parasitic smart fungi which infect grasses and crops. Then in the option D, that is Albugo. It is an obligate parasitic fungi that causes white rust disease. Okay. Then next question, in Basidiomycetes, gastroid fructification is found in option A Allomyces, option B Poxinia, option C Albugo, option D Lycoperdon. Here the correct answer is option D, that is Lycoperdon. Then what is gastroid fructification? Remember, gastroid fructification refers to a type of fruiting body in certain fungi within the Basidiomycota. In this type of fruiting body, spores are produced and mature inside the fruiting body rather than being exposed on the surface of the fruiting body like in many other fungi. So this internal spore production is a defining characteristic of the gastroid fungi. Then example of gastroid fungi include puffball, stinkhorn and some bird's nest fungi also. So here in the option D, that is Lycoperdon, is the genus of common puffball mushroom in which this type of fructification is present. Okay, then see the other option, that is Allomyces. Allomyces is a genus of fungi and the reproduction involves the formation of zoospore within the sporangia. Not gastroid structure is present. So the gastroid fructification is not a feature of Allomyces. Then in the option B, that is Poxinia. Poxinia species are also known as rust fungi, are characterized by producing different types of spores depending on the host and the life cycle stage. But Poxinia are not gastroid fungi. Okay. Then in the option C, that is Albugo. Albugo is a genus of fungi within the Oomycetes which are characterized by having sporangia and oospores, not gastroid structure, right? So here the correct answer is option D. Then next question, when two host plants are required for completion of life cycle of a fungus, the condition is called option A heterothallism, option B heterotrophic, option C heterocoelous, option D heterokaryotic. Here the correct answer is option C, that is heterocoelous. So the fungi is one that requires at least two hosts, that is the primary host in which the fungi spends its adult life and the other is the secondary host, right? So both hosts are required for the fungi to complete its life cycle. For example, Poxinia graminis causes stem rust or black rust disease, right? And it is a heterocoelous pathogen. Okay. And in this fungus, the primary host is typically wheat plant while the alternate host is barberry plant. Okay, so heterocoelous means the fungi requires two different hosts to complete its life cycle. Then see the other option, that is heterothallism. Heterothallism is a reproductive condition in fungi where two different mating types are required to produce the sexual spores, right? Then in the option B, that is heterotrophic. We know fungi are heterotrophic means they cannot produce their own food. They obtained nutrients from other organisms, right? Then in the option D, that is heterokaryotic. Heterokaryotic refers to a cell that contains genetically different nuclei within a single cytoplasm. Okay, but here when two host plants are required for completion of life cycle of a fungus, the condition is called heterocoelous. Then next question, Conidia are option A motile endogenously produced asexual spores, option B non-motile exogenously produced asexual spores, option C non-motile endogenously produced asexual spores, option D motile exogenously produced asexual spores. Here the correct answer is option B, that is Conidia are non-motile exogenously produced asexual spores. Okay, so Conidia are non-motile because Conidia lack flagella or other means of movement. So they rely on wind, water or other external forces for dispersal. Okay. Then Conidia are exogenously produced asexual spores. It means Conidia are typically found outside of a protective sac or structure. Okay, and many common fungi such as Penicillium, Aspergillus reproduce asexually through the formation of Conidia. Okay, so here the correct answer is option B. Then next question, the female gametangium of algal fungi is called option A Oogonium, option B Ascogonium, option C Ascocarp, option D Cleistothecium. Here the correct answer is option A, that is Oogonium. We know the fungal group Phycomycetes are also called algal fungi because they exhibit characteristics that resemble both fungi and algae, particularly in their morphology and reproduction. And the female gametangium is called an oogonium, right? Then see the other option, that is Ascogonium. Ascogonium is the female sex organ in Ascomycetes fungi. Then in the option C, that is ascocarp. Ascocarp is the fruiting body of the fungal group Ascomycetes. Then last one, that is the Cleistothecium. Cleistothecium is a closed fruiting body of Ascomycetes fungi. And here the female gametangium of the algal fungi is called oogonium. Then next question, gametangial copulation is common in option A Deuteromycetes, option B Ascomycetes, option C Chytridiomycetes, option D Zygomycetes. Here the correct answer is option D, that is Zygomycetes. Then what is gametangial copulation? It is a form of sexual reproduction where two gametangia fuse directly to form a zygospore, right? This fusion involves the entire content of the two gametangia. This type of copulation is common in members of the zygomycota, for example in rhizopus and in mucor. Okay, so the gametangial copulation is common in Zygomycetes fungi. Option D is the correct one. Then next question, heterothallism was discovered by option A E.J. Butler, option B A.F. Blacksley, option C Caspar Bauhin, option D Anton DeBerry. Here the correct answer is option B, that is A.F. Blacksley. What is heterothallism? Heterothallism is a reproductive condition in fungi where two different mating types are required to produce a sexual spore, and it was first discovered by an American genetist A.F. Blacksley in mucor fungi, right? You can see here that is two different mating types. Suppose blue and yellow come into contact and interact. Each mating type is also genetically distinct, right? Possessing different alleles at specific loci that determine mating compatibility. Then after fusion it forms the zygospore, and after meiosis it forms two types of spores, right? In the other hand, homothallism in fungi refers to a condition where a single fungal individual or mycelium can produce both male and female reproductive structures, allowing for cell fertilization, right? So here the correct answer is option B. Then next question, sometimes in some fungi the hyphae break up into numerous fragments and give rise to new hyphae in sugary solution known as option A conidia, option B sclerosia, option C oidea, option D chlamydospore. Here the correct answer is option C, that is oidea. So oidea are thin walled structure. They are formed when the fungal hyphae break up into individual cells, each of which can then develop into a spore, right? Then see the other option, that is conidia. We know conidia are non-motile exogenously produced asexual spores produced by certain fungi. Okay. Then in the option B, that is sclerosia. Sclerosia are hard compact like structure formed by some fungi serving as a survival mechanism and a source of nutrients. Then in the option D, that is chlamydospores. Chlamydospores are thick walled resting spores produced by certain fungi, particularly in adverse environmental conditions. But here the fungal hyphae break up into numerous fragments and give rise to new hyphae in sugary solution known as oidea. Then next question, parasexuality in fungi was first discovered by option A G. Pontecorvo and G. A. Roper, option B E.J. Butler and K.C. Mehta, option C Anton DeBerry and G. Pontecorvo, option D K.G. Mukherjee and J. A. Roper. Here the correct answer is option A, G. Pontecorvo and J. A. Roper discovered parasexuality in fungi. Then what is parasexuality? Parasexuality is defined as a cycle in which plasmogamy, karyogamy and meiosis takes place in sequence but not at a specified time or at specified point in the life cycle of an organism. And remember, parasexual cycle occurs in those fungi in which true sexual cycle does not take place. Okay. So you can see here, first the cytoplasm of two cells fuse in a process called plasmogamy. Then the heterokaryons. It refers to a cell that contains genetically different nuclei within a single cytoplasm. So in the second stage, the heterokaryons fuse in a process called karyogamy, right? Then the diploid nuclei undergo mitotic division and occasionally haplodization occurs. It means loss of chromosomes through aneuploidy, resulting in the formation of haploid cells with new genetic combination, right? So, parasexuality is important in industrial process because it allows for the development of new strains of fungi. It's also useful in genetic engineering experiment. And the Italian genetist Guido Pontecorvo first described the parasexual cycle in 1956 while studying the Aspergillus nidulans. So, here the correct answer is option A. That is, parasexuality in fungi was first discovered by G. Pontecorvo and G. A. Roper. Then next question, somatogamy is the option A, fusion of gametes, option B, contact between two gametangia, option C, copulation between two gametangia, option D, fusion of vegetative cell. Here the correct answer is option D, that is somatogamy is the fusion of vegetative cell. So somatogamy is the fusion of two somatic hyphae forming a dikaryotic cell. This is a common process in higher basidiomycetes. For example, in mushroom, the fusion of hyphae from opposite mating types resulting dikaryotic hyphae then produce the basidia. So, somatogamy is a reduced form of sexual reproduction often found in fungi like basidiomycetes where specialized sex organs are absent. Okay. Then next question, the life cycle type of budding yeast is option A, diplobiontic, option B, haplodiplontic, option C, diplontic, option D, haplobiontic. Here the correct answer is option B, that is haplodiplontic. We know the species Saccharomyces cerevisiae, commonly called the budding yeast. Then see the life cycle type. Under favorable condition, both dwarf strain and large strain cells multiply by asexual spores called budding. Right? Under unfavorable condition, Saccharomyces cerevisiae undergo sexual cycle which is haplodiplobiontic type, also called haplodiplontic type. Why? Because here both haploid and diploid phases are equally represented. Okay, so you can see here that is the haploid dwarf cells have two genetically distinct mating types or strains called positive or negative strains. Then the haploid dwarf cells of opposite strains function as gametangia and perform gametangial conjugation. It involves plasmogamy, that is the cytoplasmic fusion, and then karyogamy, that is the fusion of the positive and negative strain nucleus. As a result of sexual fusion, zygote or large yeast cell develop. In favorable period, large diploid yeast cell directly act as asci and divides by meiosis to form 4 haploid ascospores. The ascospores upon liberation form a number of haploid dwarf cells of opposite strains by budding. So the life cycle of Saccharomyces cerevisiae or in the budding yeast, here the both haploid and diploid phases are equally represented and thus life cycle pattern is haplodiplobiontic or haplodiplontic type. Okay, so here the correct answer is option B. Then next question, Fusion between two sister cells is called option A plasmogamy, option B pedogamy, option C adelphogamy, option D parthenogamy. Here the correct answer is option C, that is adelphogamy. So adelphogamy refers to a type of sexual reproduction where a fungus reproduces sexually with a genetically identical or very similar individuals, also called sister cells, that develop from the same parent, right? Then see the other option, that is plasmogamy. We know plasmogamy is the fusion of the cytoplasm of two cells during sexual reproduction in fungi. Then in the option B, that is pedogamy. Pedogamy is the fusion between a parent cell and with a daughter cell, right? Then in the last option, that is parthenogamy. It is the formation of ascus or zygote without fusion, called parthenogamy. Okay. Then next question, geniculate conidia are seen in option A Cercospora, option B Aspergillus, option C Erysipeae, option D Tapharina. Here the correct answer is option A, that is Cercospora. Then what is geniculate conidia? Remember, the term geniculate refers to a bent or knee-like structure. So geniculate conidia means the conidia can exhibit a slight curve or be slightly bent like structure, contributing to the geniculate appearance. And this type of conidia are produced by the fungi Cercospora, right? And you know, Cercospora is a genus of fungi that includes many species causing leaf spot diseases in various plants, right? Then see the other option, that is Aspergillus. They are most common type of filamentous fungi, and sometimes they are commonly called weed of the laboratory. Then in the option C, that is Erysipeae. Erysipeae is a genus of fungi, and many of the species in this genus are plant pathogens which causes powdery mildew disease. Then in the option D, that is Tapharina. Tapharina is a fungal genus within the Ascomycota that causes leaf curl disease of certain flowering plants. Then next question, which is a asexual fruiting body in fungi? Option A apothecium, option B perithecium, option C ascostroma, option D pycnidium. Here the correct answer is option D, that is pycnidium. So you can see here, it is a entire structure of a pycnidium, and it is the vertical section of a pycnidium. So it is a complex structure, globose or flag shaped and hollow. Its inner wall is lined with a layer of conidiogenous cells of various type. It opens to the exterior by a circular ostiole, right? So pycnidium is a asexual fruiting body in fungi. Then see the other option, that is apothecium. So the typical apothecium is the characteristic of the advanced members of higher Ascomycetes. It is a disc or saucer shaped or even a cup like structure. It usually varies from a few millimeters up to several centimeters in diameter, and it varies in color from pale brown to black, sometimes red, orange, yellow, or even colorless, right? And remember, apothecium is the sexual fruiting body, not asexual. Then in the option B, that is perithecium. So perithecium is a small rounded or flag shaped ascocarp, usually less than a millimeter in diameter. At maturity, it has a definite apical pore or opening called the ostiole, through which the ascospores escape. Then the osteolar canal is lined by slender, short, delicate hair-like sterile growth called the periphysis, right? So it is the structure of perithecium, and remember, perithecium is a sexual fruiting body in fungi. Then in the option C, that is ascostroma. This is the picture of the fruiting body ascostroma, and remember ascostroma is also a sexual fruiting body in fungi, right? So here the correct answer is pycnidium. That is pycnidium is a asexual fruiting body in fungi. Then last question, which spores are formed by budding? Option A arthrospores, option B blastospores, option C conidiospores, option D zygospores. Here the correct answer is option B, that is blastospores. So blastospores are asexual fungal spores formed through budding, a process where a new cell grows from the parent cell. These buds eventually detach or remain attached, forming chains or clusters of blastospores, right? Then see the other option, that is arthrospores. Arthrospores are produced by the fragmentation of fungal hyphae at points where septa are present. They are typically elongated, cylindrical or barrel shaped with a smooth surface, right? Then in the option C, that is conidiospores. Conidiospores are also called conidia, are typically formed through asexual reproduction in fungi, while they are not directly formed by budding. Then in the option D, that is zygospores, are sexual spores formed in zygosporangia after the fusion of the two gametangia, right? But here the spores, that is blastospores, are formed by budding. So in this video I cover all the 20 multiple choice question with their proper explanation. So thank you.